So for this tutorial, I'll be showing you how to draw manual ROIs for calcium imaging preprocessing. Now the importance of this step is that once we have our motion corrected calcium imaging videos, we then want to identify the pixels that correspond to cells, and these groups of pixels will be called, or are called, regions of interest, or ROIs. And this is important because we can then use these pixels that correspond to each cell and then average across the pixels to obtain time series and activity signals for each cell, or ROI. And so for this, I'll first show you how to use the program Imagej or Fiji to manually draw the ROIs, and then we'll transition to a video where VJ and I will show you how to actually draw the ROIs in the most optimal way possible. So first off, after you have downloaded Fiji, you then want to open it. So this is the initial window, and we want to import the motion corrected file. So it'll be most likely in HDF5 format. Otherwise, if it's in a TIFF format, we would use image sequence. For this demonstration, we'll do HDF5. We already already have the sima_mc.h5 file that came out of the preprocessing pipeline. So I'll double click this. This will just a warning that you can ignore. You're going to want to indicate which data set within the H5 to load. It'll be this imaging, and then press the load button. Sometimes the pixel values, the intensities, are not in scale, so you want to adjust the brightness and contrast. So you go to Image, Adjust Brightness, Contrast, and then this button will show up, and you want to adjust these sliders. Usually I adjust minimum to until you actually see something, so we can see the cell here, and we can scroll through the video, see the cells, and then what we're going to want to do is to first open up the standard deviation projection. We can also do that with a mean and max projection, as I'll demonstrate. So we'll go to Image, Stacks, Z Project, click that, and then we can indicate which frames to acquire, the start and end slices, and then for this we'll do standard deviation. We can also do the average, the max, min, so on and so forth. And this will pop out a new window showing the standard deviation projection. We can use the mouse scroll button along with the control to zoom in and zoom out. Now what we're going to want to do is to draw the ROIs. So we use the polygon selection tool. So let's zoom into a particular cell, and all you need to do is click around to the pixels that you want to include, and then to finish up the ROI, click back to where you first started, and it'll finish it. Now we want to save these, to log these ROIs. So to log a individual ROI, we go to Analyze, Tools, ROI Manager, and this will open up yet another window where we can add the current ROI that we've drawn. We can show all existing ROIs by clicking this show all. Now if we wanted to add another one, let's draw another ROI. And then add. Now you can see that. Now you can see that all ROIs that I've drawn are shown. The ROIs that we want, we now want to save them for downstream processing. And so to do this, we next want to highlight all the ROIs that we've drawn and that we want to include for downstream processing. Then click More, Save, and then rename the file according to this scheme, where first you have the file base name underscore ROI set.zip, and you click Save, and you'll see that now you have a zip file that contains all the ROIs that you've drawn. Into a recording where Vijay and I had discussed the best practices for drawing these ROIs.
All right, so this is one example session. This is one where I think I haven't, maybe I did draw it previously, but one just randomly selected session that I just saw. These are cells in the accumbens with a 0.6 mm grid length. So you can see that the grid length itself causes some aberration. So cells that are in the center are a lot crisper than cells that are outside, and so towards the periphery. So that's something that you want to take into account when you draw your ROIs. Either you could be very strict and then just like not draw anything on the periphery. I actually do draw them, but I draw them in a way whereby I'm fairly certain that it's not overlapping with other ROIs, and I can kind of show you how I do that.
Okay, yeah, cool. And I mean, I see this, is it that if you have a larger diameter grid lens, then you see more of that? Like you see that in the 1 millimeter ones as well.
So with a 1 millimeter grid lens, you'll see a lot more of a field of view that is crisper.
Okay, yeah.
I mean, it's not completely random for me to select this. I selected this primarily because I think most people use 0.6.
Yeah, okay, gotcha. And then is that a blood vessel that's running like, I guess, yeah, like along where your mouse is pointing?
Yeah, so this is a blood vessel right here.
Cool.
All these like dark spots here are blood vessels too.
Gotcha.
This one's also a blood vessel here. You see it?
Yeah, I see that.
This one's like slightly out of plane, so that's why it's like not too sharp.
Yeah.
It's actually, I mean, just a point to be made here. All of these blood vessels and just how they appear it's really helpful to have them in your field of view, because you can actually come back to the exact same field of view using those as a landmark.
Right, right. Yeah. Yeah, I've done that too before.
Yeah. It actually helps more, like, you know, when you get good at this, like you'll be able to like quickly get to these just because of the blood vessels. And then sometimes the cells are a little bit harder to align off of just because not all of them are active all the time.
Yeah.
So sometimes, you know, you'll see like a couple of cells that you think are in the field of view, but they might be like, you know, you won't know if you're off by a little bit. The blood vessels too, you won't necessarily know if you're off by a little bit, but at least you know like if you're pretty much in the correct area. And then you might need to adjust within like 10 microns or something.
I see.
The cells. So this will get you close to like 10 microns.
So over like the course of this is not super related to the actual ROI drawing, but over the course of days when you're tracking the same field of view,
Yeah.
Do you notice the cells, I guess the tissue kind of moving around / warping, or and then what about the blood vessels? Do the blood vessels largely stay in the same like relative space?
Yes, so I'll say like about 1 in 5 mice, so like let's say 20% of mice have a weird thing where they do show shifts
Okay.
um, across days in the blood vessels, and sometimes the cells appear as if they're moving too. But weirdly enough, like I actually find that the many of the same cells are in the exact same location.
I
So
see.
my feeling with those ones is that it's probably just because the lens moves a tiny bit in some mice. It's
Yeah.
not necessarily, I think, that the brain moves per se, but that the lens in some mice, it's, you know, like it's not held as tightly as in other mice maybe. And so like sometimes you see these like slight movements of the field of view.
Yeah.
My guess is it's the lens, but who knows? Like it's possible that it's, you know, the brain moving around a
Yeah.
little.
You just have to take that into mind and I guess be careful.
Yeah, I mean, like it's at least in my experience with the large Grin lenses, like the amount of movement is not significant. So like, you know, well, if you want like perfect um, alignment of field of views across days, I'd say like maybe 80% of the mice that I do surgeries on are good. Um, if you want perfect, but if you want mostly the same cells to be picked up, but practically all of them are good.
Okay, cool. Good to
Yeah.
know.
So just like something to keep in mind, I guess.
Yeah.
Yeah. Okay, so ROI drawing. So there are a few things that I want to say before we actually get to the ROI drawing itself. Um, I guess so. So what is the purpose of the ROI drawing? The purpose of the ROI drawing is for some downstream algorithm to know what are the pixels that comprise of a region of interest, right? Which is what an ROI stands for.
Yeah.
In this case, the region of interest is going to be cells, so the somata. And so in simple terms, all we need to know is what are the pixels that comprise one cell. So like let's say if you want to take this cell, you would say like the somata are basically these pixels. So anything inside that is part of the cell body. So that's an easy, quick way to know that that's basically those are the cells that you need to draw. Now, there are a few issues with that. So one issue, of course, is that if you draw this entire cell, like thing as a cell, so let me just kind of maybe draw it so you'll have a better sense of this. I'm going to zoom in a little bit.
Oh yeah, that helps. That works.
Yeah. But maybe just a side point here, like you can actually see the nucleus of the cell.
Yeah, that's really nice. Yeah.
So that shows you that this is a healthy cell.
Yeah.
So let's say we draw an outline of the cell like that.
Are you using mouse? Like a
No, this is my laptop.
that's actually not too bad.
Yeah, that's not too bad. Yeah. Thought it was going to be trickier.
Yeah.
Okay, so let's say, so by the way, just to give you a sense of how you do this in Imagej, so you just simply load your standard deviation projection in Imagej like that,
Yeah.
and you hit this one, the polygon selection.
Yeah.
You're going to use Sima to extract. I would recommend using the polygon. There's this one called a freehand selection. It works pretty well, you think, but Sima doesn't support it.
Ah, I see.
I've drawn like initially like 10 sessions without realizing
Oh gosh.
uh, that that's a problem.
Yeah. Yeah. That's good to know.
Yeah. So always stick to the polygon one.
Yeah.
Okay. And go to analyze, and then go to tools, and there's this thing called ROI manager.
Yep.
Okay, so if you pick that up, then you'll see this. So here you want to hit both of these, show all labels. So now, okay, so I lost that. So if you if you draw something here and then you click it, then you lose it.
Yeah.
So that's what happened just now. Okay, so this is where the trouble of being at home and VPNing and will start showing. Sorry.
It's all right.
I'm trying to just get this window resized.
Is that the original? That's a standard. Oh, you're zoomed in. That's why it's like
Yeah.
super zoomed in.
Yeah. This is with individual pixels now. So the problem with this stuff is
Is it possible to zoom out before you resize?
it is possible, but let me just try to resize at least.
Yeah.
Okay.
There you go.
It's a little bit of a pain to do this when you're sitting at home.
Yeah.
Okay. I think it's reasonably zoomed in, I guess. Okay, so okay, I'm just gonna leave it at that. Okay, this is fine.
Yeah, I think that's good.
All right, so now let's draw that again. So this time I'm gonna like quickly go through it. So let's say we draw this as our ROI. It looks pretty nice, I would say. So it looks nice and it covers the whole cell.
Yeah.
So obviously these pixels are, generally speaking, everything inside here is part of the cell, you would think. So you'd say, okay, this is a pretty good ROI. Now, there are a couple of issues that I think that are problematic here. So if you pick up this as your ROI, so now there's this nice thing where you can actually hide the ROIs, all the ROIs, if you just want to see what it looks like with and without those ROIs.
Yeah.
So that's pretty helpful. Okay, so now the ROI is on. So let's say your recording has any motion at all, like just any movement on a couple of frames, let's say.
Yeah.
Then what you're going to see is that this whole field of view is going to shift slightly to the left, slightly to the right, up or down, like one way, right? So there's some movement of that field of view.
Yeah.
Relative to these pixels. These pixels, remember, are like sort of fixed to your size. So like if the left corner here, imagine that as a 0 coordinate.
Yeah.
And this has a specific location in XY.
Yeah.
But relative to those specific locations, your imaging field could just move around.
Right.
And so when that moves around, imagine what happens now. You're trying to calculate the average intensity of this cell on any given frame. So that's the purpose of this ROI, right? So like on every frame, your extraction algorithm takes in the average of all these pixels and then says, well, that's the fluorescence level of my cell.
Yeah.
And then on the frames where the cells moved, then you will have, let's say the cells shifted all the way here. So it's pretty steep motion. So the cell sort of lying like that. So then about half of these pixels now become black.
Yeah, and they could be picking up like even though you can't see like other cells or processes in the kind of darker space, there still could be spurious signals in there.
Yeah. So there could absolutely be spurious signals there, but there's also, it's a darker space.
Yeah, yeah.
So it
It's
would
not the
seem
cell.
as if, yeah, it would seem as if the cell actually stopped firing
Yeah,
on an average, right? So on an average, if you look at the fluorescence, it will seem as if the cell just became quiescent.
yeah,
Um, if you see that, where you like suddenly the cell becomes quiet and then suddenly the cell becomes active again, it's likely motion. So that's basically the issue with drawing the ROI, like covering the entire cell like this. Now, of course, you can say, well, I'm going to correct the motion perfectly. And then that's fine. Like if you're actually able to correct the motion perfectly, then that's fine. But you have to ensure then that literally not even one pixel is moving in your image in any frame.
yeah.
That's hard to like be really sure about. I spend a lot of time trying to make sure that there's pretty much zero motion, at least any that I can see by eye. But even with that, like I'm sometimes not certain that there isn't any motion at all on any frame. So then because of that, you know, I find it safer to draw the ROI, not like this covering the entire cell, but instead like this, covering most of the cell, but maybe not the entirety of the cell.
Gotcha.
Now, if you do this, you know, in this case, you're generally drawing closer to the nucleus. Now, the nucleus is a part of the cell where there shouldn't be as much GCamp, as you can probably kind of see. That actually causes potentially some issues, but this is where, you know, some of the nice things about grid lenses come in, is that grid lens, like the resolution is not great, honestly, at least compared to like imaging without a grid lens and with a high NA objective. So because of that, even if you draw it around where the nucleus is, you'll still get enough of the cytoplasm in there
I see.
that you'll be fine. So you'll still pick up signals. So like clearly the nucleus is not filled here, but if you were to image using without a grid lens with a high NA objective here, you would actually see a black area here.
Right, yeah.
Where the nucleus is. Like not just a darker area, but like a black area. Then obviously you can't do this trick where you draw the ROI around the smaller region.
Yeah. So then in that case, you'd probably want to include a little bit, not all the way to like the edge, but like include a little bit more of
Yeah.
the cell, the cytoplasm.
Yeah, I mean, and this is the key thing here, is basically your imaging preparation determines the level of caution you want to apply.
Yeah.
If your imaging preparation is very good in terms of resolution, like, you know, your surface cortical imaging with very high NA objectives, then your resolution will be very high, both in XY and in Z. So now if you have any motion, you're going to be super sensitive to that.
Yeah.
In those cases, you'll have to be pretty much 100% confident that there is no motion. So that's just one thing that you have to keep in mind. Is that, so like, imagine your Z section is like about 5 microns, which is typical in tissue with like a high NA objective. Then, you know, like if your Z plane moves by 3 microns, that's already a lot. You're actually imaging a completely different part of the cell. So you'll actually see changes that are purely due to motion, not due to dynamics.
Yeah. Yeah.
That's when you know that in those cases, like it's actually very tricky. You'll have to like go the extra mile to make sure that your motion correction is perfect. In this case, notwithstanding that, I still try to go the extra mile to make sure that the motion corrects perfect. So just keep in mind that, you know, like the level of motion correction that you need will sort of depend on your question. typically because you're using green lenses, you may have a little bit of a leeway there.
Yeah.
In terms of the final, because the thing that matters here is the final output, which is basically this trace of the ROI.
Right.
And there's one thing that does affect it if you use green lenses, which is that the resolution is lower. So it means that the chance for overlap with other things is high, and we'll talk about that. But as far as motion goes, it's a little bit more tolerant towards it.
Gotcha. Yeah, that makes sense. One
Got
more
it.
question here is, what's like the minimum number of pixels you would want to include in like an ROI, like 15 to 20?
I'd say, yeah, 15 to 20.
Okay, cool.
So we'll do one example of something like that pretty soon. So, okay, so for now, this is that cell. Okay, so this is to illustrate the fact of motion, right?
Yeah.
Now, let's maybe delete that. And let me pick a different cell.
I think I know what cell you might pick.
You do? You
Yeah.
think one?
Yeah, that might be a good one.
That one's actually pretty reasonable. I think there's like one here when I was zooming in, I noticed. Yeah, okay, so let's just pick that one. The one that you pointed to. Because it illustrates the point for
Yeah.
now. Okay, so clearly this is a cell. So no one's going to question that that's a cell. Now the question is, how are you going to draw the ROI? So let's say you're going to do the original approach, which is to include all the cells, all the pixels in the cell. I'm just going to draw that here. So that's your cell. That's the area that the cell covers in your field of view. You've drawn it and added it. What's the problem with it? Well, the problem with it is that not all pixels in your ROI are from the cell. And that's the key. And what you want when you draw an ROI is that you want to know that every pixel inside that is from the cell. If not every pixel, if that is not true, then you are basically going to get contamination from other sources. And that's super obvious in this case. So this is the problem with using green lenses, is that you will get stuff like this, where you'll have a process that just goes by on top of a cell.
Yeah.
So clearly, if you were to use this and say all these pixels here are part of this cell, and because of that, I'm just going to count that as the fluorescent signal of my cell, then let me zoom out, then you will realize that your cell's activity is going to be contaminated a little bit by this cell or this cell's activity.
Yep.
And so this is a key part. Like, these are pretty far away from the cell. So there's, under normal circumstances, there's probably no way that this cell is actually going to affect the activity of a cell that's like so far away. But it does, because its process is actually going all the way to the other cell. Okay, so how will you then draw this ROI? I'm going to delete that one. So what I would do in this case is to pick a part of the cell where I see no overlap with other resolvable structures. Now, this is clearly a dendrite from that cell. All of this stuff seems to be probably some processes, but you know, it's nothing that you can resolve. So this is all going to be part of the neuropil. So then I would say, well, every portion of the cell that's above this dendrite seems to be good and is part of that cell. So that's what I'm going to use to draw the ROI. So I'll say everything that's in these pixels Yeah. So everything that's in those pixels, I think are part of the cell.
Gotcha.
And it's, yeah, nothing in there is overlapping with anything else. At least that I can see. So I'm going to say, well, that's my region of interest that measures the activity of that cell. So the benefit of this is now that you can actually avoid overlap with other things. So as I said, the benefit is basically, you get a more accurate measurement of your cell signal. The cost of it is that now your ROI does not represent the cell. So if you want to run anything like, oh, I want to know what the shape of the cells are, and maybe where the true centroid of that cell is and stuff like that, then that's not going to work here because your ROI is actually not going to cover the whole cell.
Yeah. But I think for the sake of like just functional getting the time series, then this is
Exactly.
probably the best way.
Exactly. So what I'm getting at here is that there's no single analysis that I think that will handle those two different levels of questions. One is the level of question about the function, and the other is a question about the structure. And if your question of interest is about the structure, this is not the way to go.
Yeah, I agree.
Okay, so that's that. So now let's see if we can pick up some more.
Well, I have one more question. So that the cell that we were looking at before this one, the one that has a really nice nucleus,
Yeah.
there's this. It could be a process coming off to the left. Is that actually like a dendrite, or is that some sort of artifact?
I think this is a dendrite.
Okay, it's just
It's a
super straight.
dendrite that's coming off of it and just quickly going out of field.
I see. Yeah.
So it's
Cool.
going out of focus basically.
Hmm.
So that's why you can kind of see it only for a tiny bit.
I see.
Let's see if there are more complicated cells here to draw. Yeah, maybe let's go to the periphery now.
Yeah, that's what I wanted to actually ask is how you deal with the periphery ones.
Yeah, so the periphery ones are a bit trickier. Yeah, okay, so in the periphery with a grid lens, especially with these small grid lenses, your resolution reduces quite a bit. So now if your resolution reduces quite a bit, then you start seeing things like this, where two cells are on top of each other.
Hmm.
Not just like a cell with a process on top of each other, but actually literally two cells. There's something here too, like I think there are two cells here.
Is that because the Z resolution also changes as a function of, I guess, where you are in
Yeah.
the periphery?
Yeah.
Okay.
So the grid lens has a dependence of the Z resolution with the radial distance from the center.
Hmm.
And the farther out you go, the worse the Z resolution.
Gotcha.
So I think towards the complete edges, like, like with a 0.6 mm, this is much less true for 1 mm, but with a 0.6, if you're like towards this edge, like here, your resolution is like close-ish to like just a 1 photon imaging.
Hmm.
Yeah. So 1 photon imaging, I think the resolution, Z resolution is like square root 2. So I think it's like 40% worse roughly than 2 photon.
Okay.
And so towards the edges here, you start approaching that. Now, one thing to say, like this is also true for 1 photon. So if you're using a grid lens for 1 photon imaging, that's the same thing's true. Your resolution is actually much worse towards the edges than towards the center.
Yeah.
So 1 photon towards the edge is much worse than 2 photon towards the edge. I think 2 photon towards the edge is probably comparable to 1 photon near the center.
I see.
So yeah, so just something to keep in mind. I mean, you can still it's still better, I think, than one photon, just because you can actually still see cells and the outlines of cells. But just keep in mind that your resolution's worse. So then you'll actually have Z overlap of cells.
Yeah, yeah.
Okay, so now how do you draw these ROIs? There's one approach, which is, I think I've seen maybe a couple of papers doing this, which is to just not count them.
Mm-hmm.
So if you're, towards the periphery, just ignore the cells, like just move on. I personally don't agree with it, just because, I think that that's still usable data. I think because, of course, it's not going to be as perfect as near the center, but I still think that if the purpose of this is to actually extract meaningful signals from the cells, I
Yeah.
think still use the same sort of criteria to get meaningful signals from these cells.
Just be like more stringent.
You just have to be a bit more stringent. And so this is why I personally do not use the automated algorithms for ROI extraction. I mean, I love automation, and I love coding, but the primary purpose of all of this is to get meaningful results. And so if your automated algorithms don't give you that, then you shouldn't use it.
Yeah.
And personally, I feel like most of the algorithms that exist right now are devoted towards cortical imaging without grid lenses. Most of them, not all of them. And so, and even with the ones that are devoted to grid lens imaging, I personally find that it's maybe not as accurate as I would like it to be. So, okay, so then how do I handle this? So what do I mean by not accurate? So you can pretty much right off the bat see that there's overlap between these cells. If I were to draw the ROI around the big cell, like that's probably this whole region.
Yeah.
And within that, you can probably tell that this much is actually also occupied by this other cell. And you can see this pretty clearly because you will see like an increased intensity in the areas of overlap. So like this area, like you can see that is brighter here, probably corresponds to this cell being there.
Yeah.
Okay, so how do you then draw ROIs for these cells if you can draw across the whole thing? Now, you can use the same principle. Are there parts? So remember, the purpose is basically you want to draw a region of interest where there's nothing inside that region of interest that's not part of the cell. So every pixel inside that should be part of this cell.
Yeah.
You can, I would say that if I were to draw something like this, you would probably not, let's say I'm just drawing that. I would assume that people won't have an issue when I say that every pixel here is actually part of the cell.
Yeah.
I think I can do a little bit better.
Is to go around, yeah, like to the other side, and then
Yeah. So I think I could probably go all the way here, but for now, like I would say that, like I'm pretty certain that everything here in this region of interest is actually part of the cell.
yeah, I think that's something that I would do too.
Yeah. So I'm pretty confident that that's part of the cell. So I'd use that as the ROI for the cell. So now
Gotcha.
the number of pixels here is probably about 10 to 15
Yeah.
or more than that, maybe even. Like the pixels are, I think I've zoomed, yeah. So start counting 1, 2, 3, 4, 5. So yeah, there's more than 20 here.
Yeah.
Easily 20. Right? So that's more than enough pixels that you can average to get non-noisy outputs.
Gotcha. I guess then another thing you could do is, and maybe you do this, is stepping away from the standard deviation projection and then just looking at the video and seeing
Yes.
the certain frames where the smaller ROI is active, but not the one that you just drew, to get a better sense of where that the cell above is.
Yes. So I want to say, like, every session that I draw ROIs for, I also look at the movie. Every single session.
Hmm.
Um, and that's to make sure that none of the cells that I drew or overlap, or that I haven't missed cells and things like that. So in general, if you're unsure and you want to be as close to reality as possible, then you want to look at the movie and then figure out exactly where the cells area is.
Okay.
Um, I am just in this case, I'm pretty certain that it's not as large as this. So because at least there may be processes coming out of the cell that are in that region. It's possible, but again, like, you can't see anything by eye, so it's not resolvable.
Yeah.
Right?
Yeah.
Nothing resolvable exists within that field of view, or like within that region of interest. So now, as I said, like, if you want to be less conservative and then sort of draw all parts of the cell that are not overlapping, then you would need to look at the video.
Yeah. But this suffices. I mean, that's more than 15, 20 pixels, and
Yeah, exactly. This is more than enough to get a reliable estimate of
yeah.
that sensitivity. So similarly, if I were to draw this cell, I would say I draw here. So something like that, I'd say, does not overlap with the other cell. I can't see anything else going through there, which you can verify here. Um, so nothing here corresponds to anything that I can see. So
Yeah.
I'm going to say that that's part of the cell.
That's tough to see though. The ROI2, it's actually pretty clear to me where the cell is, but ROI3, it's or the cell 3, it's I guess it's
Yeah, there's
hard to see where it is.
some element of like getting used to this. I've looked at this so many times, and not just looking at this, but looking at this
Yeah.
and comparing it to a video.
Yeah.
So many times that I know, like, the video is the true estimation of where the cell is. But I've done that so many times that I'm now like pretty good at picking it up. So I can pretty much tell that this cell is going to be like that.
I see.
And there's actually a tiny maybe thing sticking out here.
Yeah, I think there's a process or some sort of thing sticking in the middle of that other ROI.
Exactly, but I think it's quickly goes out of frame, or out
Yeah,
of, because otherwise you would see a bright spot here.
I see.
Um, I'm not 100% certain about any process going this way, so you can kind of see something going this way.
Yeah,
Because of that, when I was drawing the other one, like, I was drawing it around that, I think.
gotcha.
So yeah, see. So I stopped drawing it where I thought this process was.
I see. Yeah, I see that. I see that process.
Yeah. Um, so that's basically what you need to do. Um, it would be very useful for this to actually show you the movie and like what that looks, but I just think that that's probably not going to work with the Zoom.
Yeah, I think that's fine. I can I think what I'll do is I'll do that on my own after that,
Yeah,
and then kind of splice that together.
sounds good.
Um, but is that most of the what you wanted to say?
Yeah, I think this is it. I mean,
Okay.
the key here is to keep in mind the limitations of the recording. Keep in mind what your final output should be. What do you want to study from it? Like, remember that the most important point, I think, not just for this, but for literally like every single processing step, analysis step, is this: that your final conclusions that you make in your paper, and those are not, like, oh, this cell has this much activity here. Um, the final conclusions are going to be, oh, region A has some however many neurons that are contributing to this particular behavior or something like that. That's probably going to be the standard systems neuroscience claim. That claim should be independent of your analysis methods. That is the ultimate, you know, uh, uh, like basically qualifier for your data analysis pipeline. If your data analysis pipeline changing values there will change your final output and
Yeah.
what your claim is going to be, then you have to be super duper careful about your claim.
Yeah. Yeah. Yeah.
And in general, and this is something that maybe other people probably need to know or want to know, is that claims like, 30% of the cells are active to this stimulus, 20% are suppressed by the stimulus. Those sorts of claims are going to be super sensitive to some methods of your post-processing. Um, if you say 30% of the cells are active, I would say it's probably anywhere between 5% and like 50%.
Yeah, because you could be, depending on how you draw your ROIs, you could be getting like those false positives where you have like a dendrite that is active in a certain condition overlapping with a cell that is not, and then you would basically detect 2 times as many.
Exactly. I mean, that's just one possibility. I mean, it's not even
Yeah.
just that. That's the one of the obvious ones, right? Like where related to what we just talked about is that if there's overlap between cells, that it's going to spuriously affect that number. But it's not just that. I mean, remember that in addition to this, you also have an issue of neuropil correction.
Yeah.
And that's a whole different topic. But neuropil correction, I mean, I'll just tell you this. So if you tell me a priori that you want your nucleus accumbens cells, you want 30% of the cells to be active to a particular behavioral event, and just give me the data, I can give you post-processing parameters whereby that will be true.
Yeah.
So there's a lot of leeway here. If
There
you're
is.
not
Yeah.
going to be careful and like um, really make sure that your analysis parameters are okay, independent of the claims, and then you changing them and making sure that the claims don't change. Yeah, I mean, I think you can make pretty wrong claims.
Yeah. Yeah.
So
Yeah, I guess that's part of the reason why we're doing this is to hopefully have like a more standardized, strict kind of method to deal with these the parts of this pre-processing pipeline is that is kind of prone to, I guess, arbitrariness and has all these like variables that could affect the
yeah,
outcome.
yeah, exactly. Yeah, it's super important. I mean, the, really the only thing that I can tell you as a standard to follow is do your analysis. This is the strictest standard that you can follow.
Yeah.
Do your analysis using this method. Do your analysis using maybe some variations to your parameters,
Hmm.
and see if your results change
Yeah. Yeah. That's a good point.
in both ways, right? So
Yeah.
like maybe make your ROIs a little bit larger, maybe make your ROIs a little bit smaller.
Yeah.
Uh, See how the results change. If the results change, then you know that that's a problem. I've actually done this for like multiple sessions, not all sessions, obviously, because there's no
Yeah.
time. But it's pretty clear that they do change, and
I see.
so then you have to. That's why I like sort of evolve this more strict measure
Gotcha.
for drawing ROIs.
Gotcha. So I think just to wrap up, I think uh, there's kind of three take homes that well, two main take homes that I have from this, and then I'll add like a third one for people that I guess are starting to draw ROIs like this is to one is to make sure that your ROIs are drawn in such a way that you're really restricted for one ROI to one cell. So that means like not drawing on other processes, not drawing where there's overlap with another cell.
Yeah.
And then number two is to, as long as you have like a minimum number of pixels, you want to generally draw smaller ROIs to
Yep.
account for, I guess, motion. And
Most
then yeah,
and also any overlap.
yeah, that's true. Yeah,
Mm. Sorry. Yeah.
yeah, I agree. And then the third one that we didn't really talk about, but I think it would be good to kind of mention is I noticed that the number of kind of vertices that you drew for each ROI was around like 6 or 7. Could be more. I mean, the more you do, I guess it doesn't hurt, but you don't want to draw like square ROIs.
Yeah. Yeah. I mean, I think in general, you don't want. It's probably good to have like at least 5 vertices.
Yeah, yeah,
Just because you're like if everything is a triangle and everything is a square, it's probably not the case that all of your pixels are actually going to be comprised of that, and that you have enough pixels.
yeah, yeah.
So,
Cool.
yeah.
Well, thanks very much for doing this, VJ.
Yeah.
Hopefully, hopefully this video will be helpful for training purposes and stuff like that.
Yeah, if people have any questions, obviously tell them to reach out to me.
Yeah,
Happy to. Yeah.
gotcha.